The development of cutaneous lesions in tropically adapted beef cattle is associated with hypersensitive immune response to buffalo fly antigens. Welcome to Journal Club. The latest academic research, explained in plain English. Today we're looking at this paper from MDPI's Journal of Animals. It's authored by Nassim et al. From Centre for Animal Science in Australia, and is published open access under a CC BY license. Let's start by translating the title into more accessible language. When you read this title, you can understand it to mean in very simple terms that this paper examines how beef cattle in tropical regions get skin sores due to an overactive immune response to antigens from buffalo flies. Let's expand on that a little. The research paper explores how beef cattle that live in tropical areas can develop skin problems because they have a very strong reaction to certain proteins in the bites of buffalo flies. These flies are a big problem for cattle in these areas, and the immune system response that the cattle have to these bites is causing skin lesions to develop. This paper is trying to understand exactly why this happens and how we can help these cattle stay healthy despite the presence of these flies. Now we understand the title, which is a good start, but before we can break down the full paper, we'll need to catch up on some terminology, jargon, background information and context. Specifically, there are some key concepts you'll need to be familiar with. Let's define them now. Cutaneous lesions. Skin wounds, sores, or injuries. Tropically adapted beef cattle. Cattle that have adapted to living in hot and humid conditions. Hypersensitive immune response. An exaggerated immune response to a certain substance, causing inflammation and tissue damage. Buffalo fly antigens. Proteins or substances that cause an immune response in cattle that have been bitten by the buffalo fly. Lesion development, the process by which skin wounds or sores develop and worsen over time. Steers, male cattle that have been neutered. Intradermal injection, a method of administering a substance, such as an antigen, into the skin. Wheal areas, patches of raised, itchy skin caused by an immune response. Skin thickness response, the increase in thickness of the skin due to inflammation or swelling. Eosinophilic inflammation, an immune response characterized by an abundance of eosinophils, a type of white blood cell, in the affected tissue. Now that we understand the title and key concepts, we're ready to dive in and break down this paper. In this presentation we're going to attempt to answer 5 questions. 1. What were the researchers trying to figure out? Or more specifically, what was their hypothesis? 2. How did they conduct their experiment? That is, how did they test their hypothesis? 3. What were the results of the experiment? 4. What conclusions did the authors draw from those results? And lastly, 5. Why does this research matter? Why should you care? 1. What were the researchers trying to figure out? The researchers hypothesized that differences in immune response to buffalo fly antigens contribute to the development of cutaneous lesions in beef cattle. 2. How did they conduct their experiment? The researchers monitored Brangus steers over the course of 2 years and classified them into 3 groups based on susceptibility to buffalo fly lesions. They then prepared soluble antigens from buffalo flies, house flies, and Onchocerca gibsoni nematodes and injected them intradermally into the steers to test for hypersensitive immune responses. Skin responses were measured and biopsies were collected from injection sites. Statistical analyses were conducted to determine differences between the lesion groups and the wheal areas, skin thickness, and other variables measured. 3. What were the results? The researchers found that the development of skin lesions in beef cattle in tropical regions is associated with a hypersensitive immune response to antigens from buffalo flies. The number of buffalo flies present did not differ significantly between the groups with and without skin lesions, indicating that high numbers of flies alone are not responsible for the development of lesions. Additionally, histopathological analysis of skin tissue from animals with lesions showed inflammation and damage to the skin layers. 4. What conclusions did the authors draw from their results? The researchers concluded that the development of cutaneous lesions in tropically adapted beef cattle is associated with a hypersensitive immune response to buffalo fly antigens. They found that variations in individual animal hypersensitive responses to the antigens, rather than differences in the level of the buffalo fly challenge, determined the occurrence and severity of buffalo fly-associated lesions. They suggest that further characterization of this response could help identify specific biomarkers for breeding cattle that are resistant to lesions. 5. Why does this research matter? This research paper is important because it investigates the immune response of cattle to buffalo fly antigens and its association with the development of cutaneous lesions. This can have significant implications for the cattle industry, as it can potentially reduce the economic losses caused by buffalo fly infestations and improve the welfare of the animals. We hope you've enjoyed this Journal Club presentation of the development of cutaneous lesions in tropically adapted beef cattle is associated with hypersensitive immune response to buffalo fly antigens. If you'd like to read the full paper yourself, which we encourage you to do, you can find it at DOI 10.3390/ARNI 13122011.